You're doing. So today we'll look at past papers and questions on carbohydrate for AQA, basically for AQA. So you should pay attention to this. Try to first answer the questions. This floating tab here is the answer, so I will be revealing the answer. So just try to see if you can answer the question first, then afterwards I'll reveal the answer. Okay, so the question here says that the molecules below shows the structure of molecules found in organisms. Okay, A, B, let me use the pointer. We have A, B, C, and D. So you're meant to use this information to answer, to say, it says here, complete the table below, putting the correct letter A, B, C, or D in the box next to the statement. Each letter may be used more than once. So I want to pause the video here and see which of these, looking at their structure, each of these are organic molecules, to see which of them matches that statement. Okay, so if you've done that, I'm going to give you the answer now. So from what we just said there, B, it says it's a monomer in an enzyme active site. The idea is to show you that it's just to show you it's an amino acid. So it says a monomer in an enzyme site because enzymes are protein structures, are protein molecule, and they are made up of amino acids. So this is to show you just that this is an amino acid, basically the general formula for amino acid. It's just to show you, it's just to test your knowledge. Do you know that enzyme is a protein, and what forms the protein is amino acids, and this is a representation of amino acid. This one says D is a monomer in a cellulose, D, and that is this. Now, why is this a cellulose? Why? You know that cellulose is made from beta glucose, and that's what you're seeing here, because this OH hydroxide group is rotated already, it's flipped. That's why if it's going to be alpha glucose like this one, it's alpha glucose, but this is beta glucose. That's something you don't want to forget. Okay, so that explains why this is D. That is produced during photosynthesis and respiration. So what do you produce in photosynthesis? You produce sugar, all right, and you break that down in respiration, and that will be your glucose, and this is your normal alpha glucose, which is this one. Why is alpha glucose? Because of this OH group. Then B forms a polymer that gives a positive result in biuret. Biuret is used to, biuret, not biuret, please. Biuret is used to test for protein, and protein contains amino acid, and this is amino acid again. That's why you can have B in two places. Hope that made sense. The next question says raffinose is a trisaccharide, which means it has 3 sugar, galactose, glucose, and fructose. The chemical formulae of these monosaccharides are stated below. So each of them, this is what you have. So give the number of carbon, hydrogen, and oxygen atom in molecules raffinose. So what you're supposed to do is this. You know that this is going to join together to form a whole long chain. So the question is, how many carbon will you have, hydrogen will you have, oxygen will you have, considering all of this? So pause the video here and think about that. Please think about it, because it's not actually as very straightforward as it seems. Okay, let's show the answer now. So this is the answer. So the answer is, when you combine these in 3 places, all the carbons, that's 18. But students often want to write 12 times 3, which is 36. Like, why shouldn't I just write 36? Now let me show you something real quick. The truth is this. Yeah, you should have said 36, but don't forget that when you join molecules together, one water molecule will be formed here, another one water molecule will be formed here, which means that you have 4 hydrogen ion that will be given off. So you are forming 2 water molecules basically. So if you look at that, that means that, because it's a condensation reaction, so 2 water molecules is being formed, which means you have 2 hydrogen, 4 hydrogen, because it's 2 here, it's 2 here, and 2 oxygen. Hope that makes sense. So that's why you have, so you normally you're supposed to have 36 hydrogen. So 36 take away 4, you have 32. That's why you have 32 here. Okay. Then oxygen is basically 2. So instead of you to have 18 oxygen, you have 16 because you take away 2 from 18. Hope that made sense all the way. Okay. Would that make sense? Sorry, let me get that off real quick. Okay, so I just felt I should show you guys because most students just assume that it just be 18, 36, 18. Okay, next question. A biochemical test for reducing sugar produces a negative result with raffinose. Describe a biochemical test to show that raffinose solution contains a non-reducing sugar. So this is a 3-marker question. Think about what kind of test do you do to, if you've done a test for reduced sugars and doesn't work out, what's the next test you need to do? Okay, pause the video, try to attempt this before you see the answer. So here's the answer. So the truth is, because raffinose is basically, it has bonds between it, saying that it has, you have glycosidic bonds there, so it won't test positive for glucose or galactose or fructose, which are basically reducing sugar. So you have to break the bonds. So for that reason, you are adding HCl solution, so that will break the bond. Then you neutralize that same with sodium bicarbonate to neutralize the acidity. So now you've broken the bond. Then remember that all of this should be kept in a water bath, so to say, okay, to create the right temperature. Then Benedict solution, which is what you use for actual reducing sugar, is then added to confirm the presence of reducing sugar. Then the solution should change from blue, because Benedict is blue, to red precipitate if reducing sugar is present. Now bear in mind that you can also say it, if it say changes to green, you're correct. If it say changes to orange, you're correct. If it says changes to yellow, you're correct. Why is all of this correct? Because all of this can also be an indication of how concentrated the sugar is. So red being the brick red being the highest, so concentration of sugar. So the different color shows you the amount of sugar present in the solution. Well, that made sense. So the next question says, the diagram below shows the structure of 2 alpha glucose molecules. On the diagram, draw a box around one chemical group on each molecule used to form glycosidic bond. So these 2 structures, how would they form glycosidic bond? I think that's quite straightforward for you. Just pause the video real quickly and answer the question. So here's the answer. So this is the 2 hydroxyl group that will form the glycosidic bond. So one OH from here and one H from here. So H2O, then the O holds them together. You should have known that, I believe, or you could just check that up in the other diagrams and all. So next question. A precipitate is produced in a positive result for reducing sugar. In Benedict text, a precipitate is a solid matter suspended in solution. A student carried out Benedict's test suggests a method other than using the colorimetry that this student could use to measure the quantity of reducing sugar in a solution. So what other way instead of using colorimetry? Because what Benedict test just talks about is presence. It's a qualitative test, not quantitative. So here we are trying to say, okay, use colorimeter, please, to check for the amount. So what other way can you use to check for the amount, not just the presence? From this question, the answer is actually there. Okay, so let me show you the answer real quick. So what you can do is the precipitate, which is what was the solid matter. So how much precipitate there is shows you how much glucose there is. So the precipitate can be filtered out and dried. Then the mass or the weight of the precipitate can be measured. So that's an indicative of how much glucose is present in that solution. Hope that made sense. Okay, the Now, the next question says, what is a monomer? Just a one marker definition. Pause the video and see if you can attempt that. So a monomer is a unit that can be joined together by a bond to form a polymer. That's simple. Pay attention to this word bond, please, to form a polymer, okay? And that small unit. Next question says, lactulose is a disaccharide formed from one molecule of galactose and one molecule of fructose. Other than both being disaccharide, give one similarity as well as one difference between the two structures. So between lactulose and lactose. So that means you should be able to remember what forms lactose from your basic understanding of monosaccharides and disaccharides. Then lactulose is also a disaccharide. So similarity, one, and differences. So here's the answer. So the similarities are both of these molecules have glycosidic bonds joining them together, okay, and joining their monomers together, so to say, yeah. And the difference is that lactulose contains fructose and galactose, while lactose contains glucose and galactose. So also, let me say this to you. You can also say that this is a similarity. You can say that both of in this here, you can say both of them contains galactose because that's a similarity literally, but it's a difference really. Okay, that made sense. Please do remember to like this video if you really enjoy what you're seeing. Subscribe and share with your loved ones. Thank you. Now, glycogen and cellulose are both carbohydrates. Describe two differences between this carbohydrate. In this case, these carbohydrates are polysaccharides. Okay, so the difference between cellulose and glycogen. So pause the video here and see what you can do. That's a 2 marker as well. So the differences they have in structure. Okay, so I'm going to read the answer now. So from that, it says here, glycogen is formed from alpha glucose joined together by 1 to 4 glycosidic bond, while cellulose is formed from beta glucose and joined together by beta 1 to 4 glycosidic bond. Okay, now that's something you want to forget because of the structure. Now, glycogen molecule is branched while cellulose is made up into a straight chain. So that's basically a very big difference. Also, you can also say that glycogen molecule has alpha 1 to 4 and alpha 1 to 6. So it has 2 sets of bonds. If I've taken you this before in class. The next question says, starch is a carbohydrate stored in plant cells. Describe and explain 2 features of starch that makes it a good storage. That's a nice one. So this is not just for you to explain why starch is a good source storage molecule in plant. That's what this wants you to do. And here is an answer. Starch is insoluble, thereby not affecting the water potential of the surrounding cell, because if water potential is affected, that means it's going to suck out water by osmosis from surrounding cells. So it doesn't affect the surrounding cell because it does not affect water potential. The next thing is that, so that's why it's good to be stored. The molecules coil up, thereby making it compact for storage. Yeah, so it can stay in a small space. It coils up, so it can literally stay in a small space as well. Tick the box that identifies the test which would be used to show the presence of starch. Obviously, that would be what? Sodium potassium test, or just sodium iodide test, or just iodine test. Either way. All right, the next question talks about glycogen. It says in mammals in the early stages of pregnancy, a developing embryo exchanges substances with its mother via the cells in the lining of the uterus. At this stage, there is a high concentration of glycogen in cells lining the uterus. So describe the structure of glycogen. 2 marker. Pause the video here and see what you think the answer will be. Here's structure. Bear in mind is structure. So here's the answer. It's made up of monomers of alpha glucose. That's something you want to pay attention to. Also, the glycogen molecule is branched through the alpha 1 to 6 glycosidic bond. So it's branched, but through which bond? That's how it's branched. Then glycogen molecule has alpha 1 to 4 and alpha 1 to 6 glycosidic bond. Now, please bear in mind this. I actually wrote a 3 marker point here. You could just write 2 if you're good, because just 2 marker, but I just give an extra point. And I think I've told some of the students in class before that usually try to have what I call insurance answer. Sometimes if you can have it, please do. Insurance answer talks about you having an extra answer. Say for example, let's say the marking scheme says these 2 means the same thing. So at least you have a mark for this and a mark for this. So you still have your 2 marks, so to say. So the next question says, during the early stage of pregnancy, the glycogen in the cells lining the uterus is an important source of energy for the embryo. Now, suggest how glycogen acts as a source of energy. So, do not include transport across the membrane. You answer 2 markers. So, how is glycogen a good source of energy in this instance? Usually, students used to think this is going to be something difficult, but it's not as difficult as it seems. So, pause the video here and think about how do you think. Take note of this word energy. Where is that word again? Yeah, energy. Okay. So, because as it is insoluble, it does not affect water potential, so it can store without affecting water potential of the surrounding cells. Now, it says here, it is branched at and can easily break up. This is and can easily hydrolyze or break off to release glucose. So when this glucose is now released, what would the glucose be used for? Broken down to release energy in respiration. So see the sequence of the fact that because it's insoluble, that's really good. Even if you start from here, saying it is branched and can easily break off or can easily be hydrolyzed, please, I would prefer use the word hydrolyzed, please. Very important, hydrolyzed. So for that reason, it can easily be hydrolyzed. So glucose is released and the glucose now being broken down to release energy. Is that simple? Hope that made sense. Now, the next question is, and says, starch and cellulose are 2 important polysaccharides. So you can see the structure here, and you are expected to explain the difference in structure of starch molecules and that of cellulose. What's the difference? And this is actually a guide to see that. So pause the video and see if you can attempt that. So here's the answer to that. So starch is formed from alpha 1 to 4 glycosidic bond, while cellulose is formed from beta 1 to 4 glycosidic bond, basically. Then this position of glycosidic bond on the cellulose is of hydrogen and hydroxyl group on carbon 1 is inverted. So you can see that it's upside down here. It's upside down in the sugar molecule, because beta molecule simply means that. Just a quick reminder that if you have your beta molecule that way, so usually this is what you're going to have. You have OH here, H here, which is, this is carbon 1. This is against what you have in alpha glucose. That's just basically what I was trying to remind you there. Okay, I haven't established that. Let's move on. Now, explain the difference in structure of starch molecule and cellulose molecule. Okay, that's what we just did, sorry. Then starch and cellulose molecules have different functions in plants. So each molecule is adapted to its function. So explain one way in which starch molecules are adapted to their function. So let's look at that. Number 1 here is the fact that starch is insoluble generally. So for that reason, it does not affect water potential. Now, most importantly, it is helical in shape. That allows it to be able to coil up or to be able to be stored. That's something you want to remember. So it can coil up, it makes it to stop. Then also you can also say, I didn't add that here, that it has a branch chain which makes it easily to be hydrolyzed. The amylopectin part of starch is branched and makes it easily hydrolyzable, if you get what I mean. So that's just another answer to that. I didn't write that here. Okay. So next question says, explain how cellulose molecules are adapted to their function in plant cell. 3 marks. So you want to talk about the structure of cellulose that makes it to provide structural support to the plant, because that's what it does in plant. mainly. So it is made up of long and straight chains of beta glucose. Each parallel chains is linked by many hydrogen bonds to form a myofibril. These then help provide what? Structural strength in plant. It's possible for you to start with this sentence, this statement, that okay, it provides structural strength in plants. Okay, the next line can now be, this is achieved because it's made up of long chain, all of those points. Hope that made sense.